I'm thankful to all of you to express your interest to participate in the subgroup studies. So subgroup studies are nothing but these are the things which we do in our lab, but uh, all the institutes are not doing. Therefore, a group of centers who are doing these activities, we can collate the information so that at least a nationwide data can be available, which can be compared and which can be used. So this is the whole objective of having the subgroup studies. So, extremely thankful to the regional coordinators who have taken their interest to individually monitor their respective topics. So, I request the regional coordinators to introduce what topics they are going to handle. Parth, you can tell which topic you will be handling.
Yeah, good afternoon, everyone. Myself, Dr. Partha Gutschait. I'll be coordinating study one, ceftazidime avibactam, an astronym synergy study, and study five, that is the Vitek GNAST phone, N407 panel study.
Okay. Anand, Anand may be joining to us shortly. Dr. Pradeep, Dr. Siva is there. So, Siva, please introduce what you will be handling.
Good afternoon, sir. Sir, the study 6, we are planning to compare the cholesterol susceptibility by various methods, especially Vitek, cholesterol broth distribution method, and the cholesterol agar test. So that is the basic thing. Like there is those center who are able to do cholesterol agar test, so they can be incorporated because Vitek and cholesterol broth distribution already is happening in all the VAMR centers. So only test we are going to add up is cholesterol agar test. So if any center able to perform that, then they can participate in the study.
Yeah. So, uh, uh, Dr. Anand and Dr. Pradeep, Dr. Sandhya, yeah, I think they will join us shortly. Overall, all the centers, overall, all these six studies will be monitored by Ketan. So I wanted to reduce my burden of VMR activities. So Ketan has given his consent that he will do the overall monitoring of all the six studies. So subsequent individual meetings when we will conduct for each of these studies, the respective regional coordinators and Ketan they will be handling the individual studies. So, Ketan, you can start the session. I think 80 participants have already joined. So, I know some more will be joining. And the first study is the most important study. Many have shown interest for the first study. So, let us start the first study.
Okay,
Ketan.
thank you so much, sir. So, first study is all about ceftazidime, avibactam, and astronom synergy. And the regional coordinator for this study will be Dr. Partha. So, in this study, what is the organism group that we are going to include? So, we are going to include only carbapenem-resistant enterobacterials. Okay, and when I tell it to be carbapenem resistant, how we define carbapenem resistant in this study? So, we will be defining carbapenem resistance by resistance to any one of the carbapenems tested. Okay, and the data, the basic flow of data will come from the Vitek. We all you are Vitek users, so we are testing carbapenems in Vitek either through N405 panel, which includes imipenem, meropenem, and atapenem, or Vitek N406 panel, where imipenem and meropenem are tested. So if any one of these carbapenems comes resistant and the organism belongs to enterobacterials, then that organism can be included in this study. Now there are some three essential parts of the study. There are three individual phases of the study. The first phase of study is a must to perform AST of ceftazidime avibactam. The second part of the study is to perform AST of astronom. And the third part of the study is testing for the synergy of ceftazidime avibactam and astronom. These are three essential parts of the study. One optional part of the study is detection of carbapenems production in those organisms. That is optional, but we strongly encourage participating centers to do those. Now, what about ceftazidime avibactam AST testing? The disk diffusion methodology using a 30-20 microgram disk is what is recommended for the study. There are two strength of ceftazidime avibactam available. One strength comes as 30-20, another comes as 10-4. In this study, the CLSI recommended one, that is 30-20, is need to be procured and need to be tested, not the 10-4 one. Now, alternate other methods of ceftazidime avibactam testing like N407 Vitek card, E-strip based testing, broth microdialysis testing, those are optional and secondary. But every center which is going to participate in this study must be doing disc diffusion as a minimum testing. They can add on other testing methodologies, but disc diffusion is compulsory. We have breakpoints available for disc diffusion as well as MIC in CLSI. Then the second part is AST of Astronom. Same way, Astronom 30 microgram disc diffusion is compulsory for the participating centers to do. Additionally, they can use N406 data for Astronom, E-test, BMD, but disc diffusion for Astronom is a compulsory thing what you have to do. Coming to the third part of the study, which is going to be synergy testing. What we'll be doing, we'll be using CLSI recommended method as a compulsory method. What is broth disc elution method for synergy testing, which is recently advocated and included into the CLSI M100. When you go through the table 3D, it in details explain the process, what is recommended for broth disc elution. What you need to perform these studies, one thing you need is, you will be needing a ceftazidime avibactam 30-20 disc. Anyway, you will be procuring that for disc diffusion. Astronom 30 microgram disc you'll be using. You have to procure cation adjusted Mueller-Hinton broth. That is third requirement to start testing this test. And the fourth requirement, what I am focusing is this tube. What tube we routinely have in our setup, we have a routine test tube with cotton plug over it. I strongly discourage users of those tubes because that has a high risk of the broth being contaminated. So try to procure a screw-capped tube like this if you want to continue on this study, or even for colistin testing also, if you do CBD, then this kind of screw-capped test tube you must procure and start using this. This is a recommendation. Now, QC, you have to do ceftazidime disc diffusion QC, Astronom disc diffusion QC, and synergy QC. We'll be discussing the QC. We are restricting our QC just for basic ATCC. Like for this QC, we are restricting ourselves to the two most commonly used ATCC organisms, E. coli ATCC 25922 and Pseudomonas ATCC 27553. All the other QC we are not compulsory recommending. Same goes true for Astronom also. You just have to perform E. coli ATCC and Pseudomonas routine ATCC strains disc diffusion QC. That will be enough for us. As far as synergy QC is concerned, even if CLSI recommends testing for 4 QC organisms for the study purpose, if you use the basal E. coli ATCC 25922 for QC testing of disc elution, we are okay with it. So QC part you have to do, we are keeping the QC isolates to minimum, but those two minimum isolates you must perform QC on a weekly basis. This is very important. Now, additional testing of synergy, everybody has to do a disc elution test. But if your center wants to do more methods, include more methods of synergy testing, what has been recommended by ICMR, like E-strip based methods, then we have E-strip disc methods. The E-strip methods have multiple type of methods like stacking, perpendicular, adjacent strips, even disc stacking method. So if you want to include those additional synergy testing methods for your center, we are okay with it. But disc elution is the compulsory testing with every center must do, minimum requirement. Now coming to the optional carbapenemase detection testing, this, even if we have kept it as an optional thing, we strongly encourage centers to at least use one methodology of carbapenemase detection, because the whole purpose of this ceftazidime avibactam Astronom synergy testing is dependent on whether the carbapenemase is produced and that carbapenemase is metallo-beta-lactamase or not. Therapeutically, this is significance. So even if we have made it as an optional additional carbapenemase testing, we recommend centers to at least go for MC-MISIM testing, because MC-MISIM testing will classify the carbapenemase into MBLs and serine. And as you all know, this synergy testing has different meaning for MBLs and different implication for serine carbapenemases. So minimum you have to do is MC-MISIM. If you want to do any carbapenemase lateral flow assays like this available with is available with us, this kind of NG test carba 5 or any other ICT is available. If you want to go for PCR for common 5 CRE genes by either a closed system of test like carba or expert, or you want to go for an open platform like mini kits are available, high media kits are available, Bruker kits are available, or you have an already existing conventional PCR setup in Europe, all those things you can include in the as a part of the study. If you're going to start, try to see if you can stick with MC-MISIM testing minimum. So this will be our requirement for if you want to participate in this CZA Astronom synergy testing. So now this study is open for discussion from the and queries sorting from the all our participating centers. If you have any query regarding this, doubts regarding this study, You can ask your doubts. We'll try to address everything. So minimum testing is CZA disk diffusion, astronom disk diffusion, disk illusion synergy testing, with or without MC missing. This is minimum you have to start putting in your center.
Now this is open for discussion. If any of the participating centers you have any doubt, then you please share your points.
Excuse me, sir.
Yes, please go ahead.
Yes, sir. So this we'll be doing for true pathogens or the colonizers as well.
Ketan.
The inclusions will be clinical isolates. If you are certain it to be a
They will be all
pathogen.
clinical, no, sir.
Now, we include diagnostic isolates, not surveillance isolates.
No, I'm not talking about surveillance. Suppose I have got ET aspirate, which has Klebsiella, which is CRE, but patient is absolutely fine. So should I include that?
No. If you think an isolate to be significant enough to report to the patient, you include that in the study.
So that includes a lot of clinical correlation. So on the basis of that, if I have found out that this patient is a colonizer of this Klebsiella is a colonizer, so though I am reporting it as a colonizer, but a patient is not being treated for the same, should I include that in study or should I ignore that?
If you as a basic general rule, if you report an organism to your patient, thinking it as a pathogen uncertain or a doubtful significance or whatever, if you do a susceptibility testing for, if you found an organism to be significant enough that you put a VITEC, that you perform susceptibility testing as well as report it to the patient, include it in this study also. So that much of correlation you must be doing it before putting VITEC or before reporting to the patient. We all do. So if you find it that much significant, it may not be exact.
I can simplify the answer, Ketan. Yes. Carbapenem resistant E. coli or Klebsiella in blood, we always report to the patient. Clinical correlation is nice, but other clinical correlation we do not do. We generally we report to the patient. So if your institute has the same protocol, then you include those into the study. Suppose if your institute has a protocol that every organism you get, you go and see the patient and then decide that it is a pathogen or not, and accordingly you report to the patient. Then you follow the same protocol, and whatever you report to the patient, you include this in this study.
Noted, sir. Thank you.
Yeah, same rule applicable for antibiogram also. Our routine antibiogram, we should include only those isolates which we report to the patient. Now, how we report to the patient, it is up to us. Our clinical correlation we do by seeing the bedside, or only for a little bit of organisms like non-ferment, rare non-fermenters. For those only we do clinical correlation. So this is up to the department to decide. But if you are reporting to the patient, include in your antibiogram.
Noted, sir. Thank you.
Sir, I have a very small question. May I?
Yes.
Hello. Yes.
Yes, please go ahead.
Yeah, sir. So what we are doing is we are testing. We were testing till now by stack method. Recently, after 2024 guidelines, we have started this solution also. And we have also the ICT test lateral assay, the Carba 5 kit, and Carba R gene expert also. But what happens is not every time we check by everything. Like for example, today we had a transplant patient, so we did a Carba R. Sometimes we might go and do a ICT test. So my query is, what if we are not able to test and isolate on every platform one? And then can we have a baseline, that minimum number? Then I think that would make more sense, because it will not be a compulsion for any of the institute also, and for us also, that we have not given enough number of isolate or something like that. Because there are many platforms where we can do the testing, and it is really impossible to test on all platforms to give a comparative analysis.
I'd like to address this query, sir. Basically, what is, if you're already doing carbapenems detection by various methods, sir, minimum we need is to do any one testing methodology. Okay. The entry of this data will be taken care of by Google spreadsheet. You have to enter your data in the Google spreadsheet. In the Google spreadsheet, you'll be having an option to enter the carbapenems detection by multiple methodologies.
Okay, no, that part I understood. Ketan, what I am asking is,
for a particular isolate, suppose you are doing.
Yeah, you continue, then I'll
tell.
Yeah, please continue.
That for an isolate, whether So like for one isolate, you do a random lateral flow assay, you decided to go for lateral flow assay. For another isolate, you decided to go for a open RT-PCR or SCARBA or something. That is for the study point of view, we are okay with it. You take a call that what data you want to submit to us, what gene you detected in which platform you want to submit to us. There is no compulsion as such that you have to do it for a lateral flow assay, you have to do it with any one methodology which you think as a reliable and you perform for that isolate, you can submit the data regarding that.
Dr. Ketan, Dr. Navina from Bangalore.
Yes, ma'am.
I have a question because in case we are not able to do any of the PCR for the enzyme detection, there are some company who have promoting their lateral flow assay, this rapid carbapenemase. Is it validated?
The validation is doubtful. There currently only this NG Carba 5 is what is FDA approved. But if you are testing at your center for some other than this, if you're testing for some, this is Hardy Diagnostics. If you're doing any other ICT in your center, you can. We are okay with you can submit that data to us by submitting this. So we are not bothered with the FDA qualification or how standardized the kit is. That we'll discuss what kit you are using and how good the kit is. That we'll discuss subsequently. But as a golden rule, if you're doing some ICT and you want to submit that data, we are okay with it.
Okay, thank you.
Suppose you do multiple methodology in the same isolate, addressing to Sajad sir's question. Suppose I do a RT-PCR as well as this lateral flow assay and I get a discordant result. It is up to you what result you take as a final call and what you want to submit.
Yeah, Ketan, I'm Dr. Jayaprada from SWIMS Tirupati.
Yes,
yeah, my query is, like, if you're comparing, you are doing comparative analysis for this study, right?
Yes.
Yeah. So statistically, we have to take one gold standard and the diagnostic accuracy should be compared. And we are taking the broad, that is elution test. That's a mandatory right now.
Yeah.
As per CLSA.
Yes, ma'am.
So these are all additional tests. We can go ahead. The rapid and all these are additional tests you'd have to. But while writing in the paper, you can write these are discordant, these are concordant with the results and all. But routinely, we have to practice the disk synergy testing along with the elution test. That is the major part of our study.
Yes.
I think because while doing the statistical analysis, the diagnostic accuracy we are going to update to the CLSA. As per CLSA, we are going to prove this study. So you think about it.
Yes, why we wanted to include, that's the reason why we have made this carbapenemase detection part either by a phenotypic test or a genotypic test as an optional, because if a center is not doing, we are not discouraging that center to participate in this.
Then the isolates can be sent to JIPMER, then it can be done that there and the result can be obtained from there, I think.
That is tricky, ma'am. JIPMER doing for all the centers sending there is tricky.
No, for MSc like e-dilution or somewhere, whichever the centers are regularly doing, they can send. Instead of rapid test.
This part of carbapenemase detection, what is the purpose of doing it is the synergy has a different implication or meaning in those isolates where your metallo-beta-lactamases are picked and where the serine beta-lactamases are picked.
Yeah, but I think rapid tests are not apt and they're not validated. If we include in our study, that will become a query from the reviewer. Like for vision, I'm saying
we can take a call on this rapid test feasibility and the reliability. We can take a call on this when we
can do it, sir. But to include in the study, additional we can send in the Excel that results and all. But inclusion in the study is doubtful, sir. My suggestion that's all.
Yeah, sure. We let us have all this study will be individually discussed in a stretch. So at that time we will discuss about this point, and we have the last day of submission is 10th. So after that only all this individual discussion will start. So we have 4-5 days to go through the literature, go through the literature and get back to you. Okay.
Yes. Thank you. Thank you.
Sir.
Yeah.
Sir.
Yeah. Please go ahead with your query.
Yes, sir. Sir, actually, how many isolates we have to do, sir? Is there any minimal number?
We have
for statistically significant minimum 30, sir. At least 100 or more. To evaluate to do the statistical analysis, sample size should be 30 for this. So more than 100 if you submit, if that is it is good study.
Sir, here small suggestion here. See, the disillusion has been just rolled out by CLSI. So if we do not hurry, I think the value of the publication would actually decrease if we keep it for a very longer period. So what we were thinking is that we should actually have the study for maybe 2, 3, 4 months depending on the number of isolate, and wrap the study up in maybe 8 to 10 months, so that by the time the new CLSI comes, we should be having that publication, if possible, in hand.
Yeah,
I think that.
Sajad, your point is well taken. When we have individual meeting, we will discuss about the sample size. Okay.
Yes.
Okay,
Yes,
sir.
sir.
Okay,
So
thank you.
see, this meeting is for us also to gather the questions of the participating centers. So whichever we are not able to address at this point of time, definitely in the individual meeting we'll address. Okay. So
Okay, sir.
sample size we have never thought to. I mean, we have never discussed about this. So yeah, it's a good suggestion that instead of having a 1.5 year of study, we can have a smaller study. So we will discuss about this subsequently.
What is required out of centers is once you start participating in this study, all the carbapenem resistant enterobacters what you get, you must undergo this study, the pathogenic ones. But
Yeah.
it's not that I selectively did it on some days, then I stopped doing in between, then I again started doing
See,
it. This is what is strongly
sample
discouraged.
size and study duration can be discussed, but whatever sample size and study duration has been fixed, will be fixed. You all have to adhere to that, and continuously all the CRE isolate you have to include. Randomization will have a negative impact on the outcome. Randomly doing on the CRE isolate. It should be continuous selection of CRE isolate. Okay, I will show you the list of the centers who have expressed interest in this study. Yes, any other query?
I have something, Apurva.
Yeah, please, madam.
So the thing is that probably I've missed many things. I wanted to know what exactly were the objectives, and this is the sub-study of your VAMR study, is it?
Yes, ma'am.
Okay,
Yes,
because when we, for us, no, any sharing of data, or for many others probably, we require approvals, right? And nowhere in the primary study we have mentioned anywhere on these things, because the primary study was basically all Vitek and Vitek isolates. So this study is all on disk diffusion. So I don't see the correlation between the study objectives and the original study objectives for what we have participated. So for me, more than a sub-study, it will become a new study.
Yeah,
So
but we don't want to consider these as new studies because our study title is now changed. It is AMR surveillance, quality strengthening of AMR surveillance by using automated AST system. And in the protocol, it is discussed that whenever automated AST system is not available, or requirements are not, or the methodology is not available, we have to reconfirm by doing a secondary test. So by taking this broad objective, we are enrolling the individual studies.
Okay.
If some centers it is difficult to convince their management that it is the same study, then it will be difficult, ma'am. We will not take it as a new study because it is going to be so difficult to apply for ethical clearance and all that again.
No, so for the same logic that you are holding, that this is the name has changed and therefore you have broadened that. So I have to use that same thing and say that, okay, this is the main, now this is the study with the changed name and it includes all these things. So for the same logic you're giving, I have to give them that same information. So that information, whatever, if you have something to share, no, then that will be useful for us to say that, yes, this is the study and these are the subgroup studies which we would like to take part in.
Yeah, that we can do, ma'am.
Okay.
That we can share a mail to you, and whichever subgroup study you want to participate, you can enroll.
Okay, so that objectives of this particular subgroup study, is it available somewhere, or
we
you
have
have
SOP, ma'am. We have SOP like a write-up we have for all the studies.
Okay, so that also if you could share whenever you all are sharing, so then we put that up. Then you all have said that any isolate of which is carbapenem resistance, right? So then we'll have to do this. So that's not an issue. And so my main concern was this. The other concern was that now we are not doing the MCIM methods. We were basically doing for e-test, but we were not doing, but we are e-test plus the disk. So, but we were doing it only for because of a project. Now, if this has to be done throughout, because e-test has its own cost limitations. So, because this project was funded, we were able to do that. Now, if it is something by broth dilution or for the disk elution method, which is reasonably less expensive, then we'll need to validate that method. Now, to validate that method, if there are any particular strains that you have or a QC that you can give for us to validate, then that will be useful for us to do to include this or to validate this method to start getting involved in the study. We are not doing any rapid lateral flow assays, mainly because not validated, and we don't have controls or we don't have specimens which have said that, okay, this has been tested by this method and therefore I can, and that will require large numbers because there are so many different kinds of enzymes and methods.
Yes, ma'am. Lateral flow essay, anyway, we are now debating. So, because some of the participating centers have expressed their negative comment on this. So, when we will discuss this study in detail, that time uh, we will have a discussion on whether to include the lateral flow study or not.
So my question was for the elution method. So you have said for the other methods to use the E. coli and the Pseudomonas. Now for this method, is there any other specific strains that we need to use?
There are total four ATCC strains, what reference strains, what CLSI recommend, ma'am. The first what we commonly use is a non-CRE strain, that is E. coli ATCC. Other than that, the three strains have specific resistance pattern, which is recommended. The problem is procuring those strains will be a thing. If you go through the table 3D, you'll see a list of four ATCC strains. We currently don't have those strains, as well as we are not making it compulsory for institutes to buy that for all the three other strains. But if your center can procure those and start testing, that would be the best thing. Because other than E. coli ATCC, which is a non-CRE strains, there are three more strains recommended by CLSI, which I have shown in the PPT. It's given in table 3D.
Okay, then how do you ensure the QC of this method across sites?
The quality of, ma'am, we are ascertaining the quality of the disk what we get from the, and by doing individual CZA and ATM QC, disk QC, and the method will be tested by at least E. coli ATCC to make sure that the illusion and all errors can be picked up.
Parth, okay. you want to add something, Parth?
No, sir.
QC.
Like Ethan said, we are checking the disk quality with the recommended ATCC. As per the CLSI, we have kept only the minimum thing like ATCC, E. coli ATCC 25922 and Pseudomonas ATCC. We are not going for a BLBLI specific ATCC as recommended by CLSI to decrease the burden on the center. So those are the minimum things which we have kept as a compulsory QC, and we have not kept Klebsiella 21461705. Those are mainly for OXA and NDM specific. So if your center can buy those, that is welcome.
But you have suggested cation adjusted MHB or MHA. Yeah. So those cation adjusted, whatever ATCC strains you are talking about is only for the disk. But for the media, that is a cation adjusted medium, we just go by the enterococcus with cotrimoxazole.
Yes, ma'am.
That is
any other specifications?
Ketan, enterococcus cotrimoxazole is good enough, no?
For that is not for this
MHA.
MHA thymidine content. For there is no specific ATCC available to look into the quality of CAMHB. The overall process control can be used. Like I told, if your CAMHB is inferior, if your disk is not eluting properly in those disks we use for disk illusion, CRE strain will have a erroneous result. So even if you do with that, if your process is wrong, then that will be picked by one ATCC. What is more accurate is if you use a specific genetic marker ATCCs, then that will be an additional burden for the participating centers. That's why we don't want to make it. It's anyway, centers are encouraged to do those, but as a compulsion, we don't want to put a pressure on the.
I think the CJD remaining discussion we can have in the individual meeting.
Okay, thank you.
This meeting is to have a superficial discussion so that the center will be aware of what they are. Many centers are confused whether they are eligible to participate. So we can have always a discussion on this, ma'am. So
yeah, thanks, Apurva. Just for me, it is the first part of it, to convince.
Yeah, ma'am. I know you have a very stringent protocol. Okay, I'm just showing you the list, all the participating centers. So these are the centers who have expressed interest on the first study, on the CZA study. These many centers have expressed interest. Some of them have filled twice the Google form, but these are the roughly it is around 16 centers. 16 centers have shown interest in this study. As of now, the last day to fill is still there. So as of now, this many centers have expressed their interest. So Ketan and Parth, thank you for your designing of the study. So Ketan, you can go ahead with the next study.
Yes, sir.
And we have to be quick because 1 hour we have spent and only one study we have discussed. Detail of methodology you don't discuss, Ketan. Just discuss what the centers are supposed to do.
Yes.
Whether they are.
Yes. The next study is our Vibrio AMR surveillance or antiviral study, which is primarily based on disk diffusion. In this study, we are sticking to Vibrio AST surveillance by disk diffusion methodology. Specimens you can include is all the specimens wherever you get a Vibrio isolate, Vibrio cholerae, as well as non-cholera Vibrio species. The identification of Vibrio For that, we have a list of conventional testing that will share subsequently with you. So conventional testing plus minus anti-rust testing for Vibrio cholerae and conventional testing for other Vibrio is okay with us in this study. Automated with Vitek GNID card plus minus anti-rust testing for cholerae is acceptable. Anti-rust testing is preferable for Vibrio cholerae, but if you don't do it, still if you stick to your conventional testing very stringently, then we are okay with this. The testing methodology will share in the SOP subsequently with you, but all the biochemicals we included are the routine ones, which we use routinely in our laboratory. We encourage centers to do, if you isolate Vibrio cholerae, we encourage isolate institutes to do the biotyping. But if you don't do it, we are okay with it. But we still encourage you to go ahead with the biotyping of Vibrio cholerae. Regarding the AST in this study, this diffusion is compulsory. Okay, and MIC-based testing using BMD or Vitek, this is optional. As you all know, Vitek is recommended only for testing for non-cholera vibri. So if you do that is a supplemental testing. Every participating center must do this diffusion as a compulsory method of AST testing. This is the main part of the study. And how we do that, what antibiotics to be tested. So we'll create a Vibrio panel, dedicated panel for Vibrio cholerae and dedicated panel for non-cholerae Vibrio. And all those antimicrobial list agents will share with you, and you have to do testing for those by this diffusion. Entry of data will be done straight away for IBER platform. Like I told for the previous study, you have to do the entry through a Google spreadsheet, share to you as an individual center. But here in this Vibrio study, the data entry will be done through the IBER platform, as we routinely do for other isolates. You just have to enter the ID of the Vibrio, a panel will appear, and those panel data of this diffusion plus minus Vitek you can enter. So this is about the Vibrio study, the identification of Vibrio as well as the AST by this diffusion. The panel will share you. We have the panel ready with us. We will share you all our common antibiotics recommended for cholera.
And Ketan, Vibrio is a very rare organism. Every center may not have Vibrio. So whoever will express interest, if at the end of the study period, if they do not have adequate no isolate at all, zero isolate at all, then that particular center cannot be able to continue in the study. So because we all are not aware of whether we'll get a Vibrio isolate in future or not. So in a year we will have very few number of isolates. So in fact, I would urge that all the centers should participate in this study, because there is nothing additionally to be done. Only you have to put disk diffusion. I mean, why the purpose of keeping this study as a subgroup study is we are doing disk diffusion for it. We are not doing Vitek for this. This is the only reason why it is kept as a subgroup study. Otherwise, Vibrio data collection is a part of our main study itself. So, in fact, I would request all the centers to enroll for the study. And if they do not have Vibrio isolate at all in the whole study duration, then ultimately their center will be excluded from the analysis. Ketan, doesn't make sense because because only 3 centers have filled the form, and all the 3 centers have told that their yearly isolation is less than 5. So in that case,
that is the reason of this study, sir, because none of the center can generate their Vibrio antibiogram using 4 or 5 annual isolate. But if you pool 10 such isolates, we have a good 50 isolate, and those all the centers can use that Vibrio antibiogram for their purposes. So
yeah, so maybe Anand, you can speak to all the centers, and you make this this particular study applicable to all the centers. Let everybody enroll for this study, and if they do not have Vibrio at all, then can be excluded from the study at the end of the period.
Okay, fine.
Yeah, otherwise, if only 3 centers will enroll or 5 centers will enroll, then we will again end up in having no data on Vibrio. The phase 1, the whole year is gone, but in phase 1 we were having 0 data on Vibrio. When we have done an analysis of that, why we didn't have a single isolate of Vibrio, we realized that it is a Vitek based study, so Vibrio only distribution is done, so centers would not have filled their data. That is why we have created this subgroup study. So let us have all centers be a part of the study, and we can have the Vibrio. Let us have a vision that at the end of phase 2 we will have an antibiogram of Vibrio, which will be again a dream come true, because such a rarely isolated organism antibiogram is very difficult to achieve. Next study, Ketan. So Anand, you were not there at the beginning of the session, so can you just say a few words? How what is your plan about this study?
Yeah, thank you. Good afternoon, everyone. Myself, Dr. Anand Janapur. I am from Mayasnizingappa Medical College, Bagalkot, Karnataka. As Dr. Apurva has already reiterated, the purpose of these studies are not different from the main study. Here again, we are doing AMR surveillance specific for various species in this sub-study. And this meeting is specifically for orienting individual centers so that they can decide whether the additional tests, if required, are feasible or not, so that they can make a decision to. So as far as this study is concerned, the requirements are very simple. Most of the labs will already have everything. For identification, basic biochemical test, serogrouping with O1 anti-sera, and biotyping, simple antibiotic test, that is polymyxin B, is enough. As far as AST is concerned, because we are doing distribution, that should be feasible. So I encourage all the centers who are participating in this meeting to be part of this study also. Thank you.
Yes, Anand.
Yeah.
Yeah. So
Over to you.
Yeah. So let us hope that at the end of this study, we will have some biblio data with us. Yeah.
So we were talking about rare or isolates, rare organisms.
So Ketan, you can finish Haemophilus first. Sandhya will be joining shortly. So you can you can finish Haemophilus first, then we'll go back to.
So all these subgroup studies are for those organisms which we or any other center individually don't get sufficient number of isolates so that they can have their own institute specific antibiogram. If we pull up the data and take a little extra step towards it, then only we can have a pulled up antibiogram which can be utilized by all the centers. So on the same way of discussion what we have, this study 4 is all about Haemophilus. And again, for the ease of implication, we are keeping it as a disk diffusion based study. We can include all the specimens from wherever you get a Haemophilus isolate. How we recommend to identify Haemophilus? Using automated based ID is compulsory for Haemophilus. You can use either VITIC card, that is the NHID card, Neisseria Haemophilus ID card. You can use other automated ID platforms. If you can use Malditoff, but including automated ID for Haemophilus is going to be compulsory for you. What about conventional testing? The conventional testing can be utilized as a supplemental testing because sometimes automated systems also have their loopholes in identifying Haemophilus. We encourage people to use growth on chocolate and see blood agar, colony characteristics, gram stain, oxidase, and all those things. Demonstration of satellitism is going to be a compulsory test you will be putting. It is given positive by some species of Haemophilus only, but testing for satellism is going to be a compulsory test. You must do identification automated, optional conventional, and must do satellitism test as a routine for any suspected Haemophilus isolate you get. Factor disc based testing, that we have factor VX and XV discs are available. And if you want to do those testing in the backdrop of tryptic soy agar or nutrient agar, that is optional. But if you do, then it's a good thing because that will help you identify more accurately the species of Haemophilus. So that was about the ID of Haemophilus. The AST, the media what we recommend is strictly abide to Haemophilus test media. You can have an in-house prepared STM media. How to prepare it is given in CLSI, very nice and clear. The base of that is Mueller-Henton agar, or you can buy a commercial STM media available, but you must do the testing on STM media. Testing disc diffusion on chocolate agar is strongly discouraged and is not recommended in the study. So procuring your STM media, either preparing in-house or getting commercial HTM is optional to both to the centers, but HTM is must. Now, the problem with is breakpoint that Haemophilus influenzae, the CLSI breakpoint is only dedicated for influenzae and para-influenzae. The HASIC breakpoint we have for all the other non-influenzae, non-para-influenzae Haemophilus, but those are MIC-based breakpoints. So M45 breakpoint is not going to be helpful for us. For the sake of study, if you get any Haemophilus isolate, which is non-influenzae, non-para-influenzae, just for a trial basis, we can use CLSI M100 breakpoint and take the data, interpret the susceptibility. Subsequently, we'll see how much of non-influenzae, non-para-influenzae isolate
very
we get.
Rarely. Very rare, Ketan.
And
Most
then
of the isolates will be influenza, para-influenza only.
Yes.
So very rarely you may have some other species. So that is okay. We will we'll follow M100 and we'll finish it up.
Yes, we'll use CLSI M100 data, and the list of antibiotic will be, the data entry will be through IBAR platform. So like you have used IBAR platform, the organism need to be named to be entered.
Again, Yeah,
Panel.
again for hemophilus, the rule is, I mean, the participation rule is same. What we have discussed for our Vibrio, all centers should enroll for this study because hemophilus isolation is a part of every center's routine exercise. Why it is kept as a sub-study? Because we have used disk diffusion as the ASD data collection platform, and that is why it is kept as a sub-study. So all centers are encouraged to enroll for this study. If they don't have hemophilus isolation at all in the study duration, yes, in that case, that particular center will be excluded from the analysis. But enrollment should be encouraged to all centers. All centers should enroll themselves. Yeah, but one thing extra in Vibrio, 100% all center can enroll. Here, because HTM is asked, like hemophilus test medium, we are asked. That is why if some centers are not, this is the only difference between Vibrio and hemophilus study. In hemophilus study, we are asking hemophilus test medium as a additional procurement to be done to perform the study. So, but the centers should take this as an opportunity to implement hemophilus protocol in their lab, because hardly one HTM packet they buy, one box of HTM, it will go for 5 years or even 10 years. So it will not be a waste for them. So this study can be used as a platform, as a opportunity to implement hemophilus protocol in their laboratory. Right, Ketan?
Yes.
So Pradeep is going to look after this study. So Pradeep, please introduce yourself and how you are going to monitor the study.
Yes, yes. Good afternoon, all. Myself, Dr. Pradeep. I'm currently working as assistant professor in Department of Microbiology, AIIMS Nagpur. The main focus of this study will be first to encourage all centers to perform proper SOPs regarding how do we isolate hemophilus, because many times we tend to miss, especially in sputum sample, where it is recommended to place becidracin disc as well as the staph streak, which is needed.
So we will have a separate session, Pradeep, when we have the
yes.
hemophilus protocol discussion, no, only for those centers. Those centers,
We'll have
we are encouraging all centers to participate. So we will have a separate protocol discussion of hemophilus influenza where the entire isolation protocol we will discuss.
Sure.
And I will try to request Dr. Sujata from JIPMER to be present. See if I could convince her, it is nothing like that, because he is extremely experienced in isolating hemophilus influenza.
Yes, sir. Yes, I would like to take this opportunity because I had worked with ma'am for initial 8 months of my SRship, and I got to learn so many things from ma'am only, sir. Yes, I accept.
So I will try to request Dr. Sujata, and we will take a slot from her, and you can present, and madam can moderate the session.
Yes, sir. Yes, sure.
Yeah.
Thank you, sir. Thank you.
So here also, Ketan, please encourage Ketan and Pradeep, please speak to all centers. Encourage maximum centers to enroll for the study. Okay.
Yes, sir. Yes.
Only when they will tell that we cannot afford hemophilus test medium, only those centers can be excluded from the study. Otherwise, enroll as many as possible. And in the protocol discussion session, because isolation protocol you are going to discuss, even if those centers who have not agreed to be a part of the study because HTM they cannot buy, even if those centers also you can ask to attend the session because hemophilus isolation protocol they will learn. Okay, so
yes,
that's
sir.
so much of gain to them.
Sure, sir.
So the next study is Burkholderia pseudomallei study. So this is also a very interesting organism where it is some states of India, especially south zone of India, have a good load of Burkholderia pseudomallei. Unfortunately, this organism also the MIC breakpoint is only available by CLSI, and Whitech doesn't have approval for Burkholderia pseudomallei. Therefore, disc diffusion has been suggested by the pseudomallei group. We have discussed with Dr. Chiranjay from
KMC.
has advised us to follow UCAS guideline, because now UCAS is available for last 2-3 years, where both disc diffusion and MIC breakpoints are available. So he has advised to go with disc diffusion. So Dr. Sandhya is going to monitor this study. Sandhya, if you are available, then you can summarize the study, and you and again, this is not a subgroup study like Vibrio and Hemophilus. Even Burkholderia also is not a subgroup study. It's like Burkholderia isolation is going on for all the centers. Is that we are only that we are collecting the distribution data, therefore we are keeping this as a subgroup study. Otherwise, all the centers across VMR are requested to enroll for this study. Study 2, 3, and 4. Vibrio, Bacillus, Pseudomalai, and Hemophilus. Let all the centers join. If they don't have any isolation at all in 2 years, okay, that time those centers can be excluded from the data analysis. Sandhya, you are present?
Yeah, I'm there.
Yeah,
Yeah,
so
good
you can.
afternoon all. Am I audible?
Yes, yes.
Yeah, coming to this analysis of Burkholderia pseudomalai antibiograms. So here we encourage all the centers to from all the specimens the isolation of Burkholderia pseudomalai to be data to be entered, and here identification point of view, because the identification by conventional method is very difficult. Just even though presumptively we can identify based on colony characters, typical colony characteristics, and by the colony gram staining. But still, we recommend strongly, the study team strongly recommends to use Vitek gram-negative ID card for the identification. And in addition, if centers are using PCR or other ICT methods can be considered optional, but compulsory for ID is the using gram-negative ID card. And coming to the doing susceptibility testing for these isolates, pseudomonas isolates. So as previously told, so here, because the UCAS guidelines it gives the recommendation both for disc diffusion as well as for the MIC guidelines. So, but it is available MIC. There is no platform by which we can test all these drugs in one platform and give the interpret the results. Because in Vitek there is no drop down box of for Burkholderia pseudomalai. We cannot take the printout for the MIC reading. That's why we strongly recommend all the centers when you isolate Burkholderia pseudomalai to do disc diffusion and the guidelines we will share with all the centers, how to do the methodology and all, what and all the disc, antibiotic disc to be tested. So all those will be shared and uniformity to be maintained for all these isolates. So important point here to remember is identification is compulsory by a Vitek gram-negative ID card, and AST is by disc diffusion method. And the drugs, whichever antibiotic agents to be tested, that we will share the methodology will in future shortly.
Yes, Sandhya. So
yeah, so isolation will be maybe it is limited to some of the places, like it is not it may not be very common in north zone or some of the zones. So but still we recommend all the centers to enroll. If you don't get any isolates in this 18 months, then you will be automatically excluded. But otherwise, as and when if you get a suspect Burkholderia pseudomalai isolates, kindly do GNID testing by Vitek, confirm the ID, and then do the AST by the disc division and interpretation by UCAS guidelines.
yeah, so till now around 8 centers have enrolled for pseudomalai study,
They are
and
from
5.
which area? They are from the centers.
One is JIPMAR itself. Other are Velaman Institute, Aster Hospital, Kim's Kingway Hospital, RCC Trivandrum, Apollo Bhubaneswar, JNU Aligarh, and DY Patel Medical College. So these many centers have enrolled, but as yeah,
all the centers to participate in the study.
Yeah, we can we can urge all the centers to participate in the study. In fact, if they attend the first session, the study protocol session, the isolation protocol also, they can understand better.
Yes. Yeah.
Okay.
Anything else?
Any doubt for if you have any query for Hemophilus?
Say medical college there. I have a small query. Actually, our sample, what we have enrolled was blood and sterile body fluids. So with that, am I audible?
Yeah, you're audible.
Yeah. With that, I think hemophilus is possible. Vibrio, we cannot able to. Is it okay?
So I want to tell you that for Vibrio, Pseudomalai, and Hemophilus, please don't consider the sample what you have enrolled for Vitek. Okay, those Vitek based data, we have you would have enrolled blood and sterile body fluid. But for
Yes, sir.
Vibrio, Hemophilus, and Pseudomalai, let us consider across all samples, whatever isolation you have, you have to put disk diffusion and you have to enroll in the study. Okay, clear?
Sir, one small,
Fine, sir. And
so
one
one
more
small,
thing,
Yeah,
sir.
yeah,
Yeah,
yeah. Please
one
clear
more
it out.
thing. Actually, yeah, we have more of salmonella isolates. Maybe if like, shall we include that as a subgroup study, sir, or
Yeah.
many centers are getting
Salmonella, why we have not
salmonella more?
considered as a subgroup study? Because Salmonella, we are supposed to do Vitek and Vitek AST, and after that we will be enrolling in the study. So Ketan, can we extend the same concept for Salmonella and Shigella also?
Enteric pathogens, yes, sir. We can do that.
Yeah, all the diarrheal pathogens we can extend the same idea, because if they don't do Vitek at all, if they are doing only distribution, it is acceptable to include Salmonella, Shigella data also in addition to Vibrio, isn't it?
Because anyways, the enteric salmonella and Shigella isolate, the antibiogram is often weaker for an institute because of this reason, the less number of isolations. So if we can include in that, then we can have a robust antibiogram for them. I agree with.
Yeah.
We many enteric salmonella species from our. So that's the reason even I was thinking to ask you.
Yeah. So I actually your question is very valid, and it created another opportunity for us to extend the study for Salmonella and Shigella also.
Yes.
So all the diarrheal pathogens, Salmonella, Shigella, Vibrio, Haemophilus, and Bacillarius pseudomallei. These many organisms we will go for distribution, and across all the specimens, whatever isolation we get, we will include them in the study.
Thank you, sir. And one more, this one, sir. Sorry, taking longer time. Like as we discussed, for the first study, there are a lot of ATCC strains we needed. Can we just see whether yes, sir, any of the center is having ATCC, that kind of strains already?
First study,
Then
ATCC
we
strains
can
are the
pool and
routine.
distribute among ourselves.
First study,
Carbapinamase detection, sir.
yeah,
Like
yeah, CJD study, the ATCC strains are the routine studies only, routine QC strains what we use. We have not, we have not added any extra QC strain which has to be procured. Okay, so with the routine QC strain only you can manage the first study. So you need not be worried about that.
Thank you, sir.
Yeah.
Hello, sir.
Yeah.
Sir, we have one doubt on Haemophilus, sir.
Yeah.
Sir, Haemophilus agar you said no, sir, to purchase. Sir, even though if we purchase, we don't have 5% CO2 incubator. So can you suggest any other, sir?
Ma'am, yeah, if ma'am, we can proceed with candle jar because that will be able to give from 3 to 5% CO2. So in that case,
sir.
candle jar can also be used as an alternative for 5% CO2.
Okay, sir. Okay. Thank you, sir.
Yes.
Yeah. Pradeep, you can find out the rate of Haemophilus test medium from High Media.
Yes, sir.
And,
So commercially available HTM usually it costs approximately 120 per plate, sir.
120 rupees per plate.
Per plate, yes.
Okay, that is a commercial medium. And can
Yes.
we procure the powder and prepare ourselves?
Yes, sir. Yes. Uska cost I will find out, sir. All the consumables which are required to prepare in-house HTM, that cost as well as its exact preparation
Yeah,
method we can discuss.
the local high media you can find out, so that at least the participating centers will have an idea that how much they are going to be.
Yes,
and
sure,
that
sir.
should not be a reason of non-participation, because
Okay.
Haemophilus isolation is a part of everybody's lab. Once they isolate Haemophilus, they have to put AST. So
Yes, sir.
to put AST, they should have HTM.
Yes,
So
yes.
if the HTM is not very expensive, then every center can participate in this subgroup study.
I accept, sir. Yes.
So next 1 week or so, we will have, I mean, after 10th, the last date of submission, the following 1 week, we all will have a discussion on individual protocols. So one day Haemophilus, one day Pseudomalai, one day Vibrio. So each of these protocols will be discussed. So that time, maybe all these small queries we can address. Okay, we'll go to the next study. Parth and Ketan, go ahead.
So next study is for those centers who are doing or willing to do testing on a specific gram-negative AST panel of Vitek, that is N407. Currently, what we are doing is in our VMR study, we are excluding testing by 407 primarily because of its non-uniformity of mode of testing. We are not considering 405, 407 testing as a part, but we thought that if there are some specific centers who are doing it in a fixed way, fixed pattern, then maybe we can do a subgroup analysis for those centers who are doing Vitek AST and 407 testing. Now, what is the inclusion? So any gram-negative isolates obtained from any of the clinical specimen can be included, but it must follow some specific rules. Now, it is the discussion among us and the participating center which is going to finalize the rule. But some of the acceptable rules which are acceptable for including as an anti-biogram analysis, if you do a selective testing from from a particular group of antibiotic, like for example in our case N407 panel, would be if suppose you include all the blood isolates, all the gram-negative bacteria you get from your blood isolate, you do a N407 testing, irrespective of you are doing it testing by 405 or 406. All ICU isolates, if you get, and you include it testing by N407, these are acceptable selections. Suppose some particular department of your institute specifically asks for 407 testing because they want to use those antibiotics in their patient. For example, medicine department or any particular department asks for it. If all the gram-negative isolates from those particular department you test by 407, those can be included. If you have a fixed pattern that all carbapenem-resistant isolates, you always do 407, not selectively. Some I did, some I didn't do. Not like that. All carbapenem-resistant isolates, if you do a testing by 407, that is still a acceptable pattern. But you should not be selective on those categories. You should not select some CR isolates and leave all the CR isolate for 407 testing. Or any fixed combination of these, either a specimen-specific combination, ICU department, or resistance pattern, like most common is carbapenem-resistant isolates, should be decided. What is not recommended is you decide if you are having an XDR organisms. So you have a carbapenem resistant organism for which amikacin or maybe cotrimoxazole came susceptible. Then you stopped doing it 407. If everything comes resistant, then you did it. This kind of random selection is not recommended based on the resistance profile. Sometimes you did, sometimes you didn't do. Is not at all recommended. Then you cannot participate in this 407 study. You must formulate a very specific kind of pattern like these. And we can streamline it once we have a discussion with all the participating centers. But on an average, these four are the filters. These four are the rules what applies for 407 testing: blood isolate, all ICU, a particular departmental isolate, or all CR isolate, irrespective of its susceptibility testing to other antibiotics. You don't need to look into other class of antibiotic whether they are susceptible or not. If it's a CR isolate, you must do a 407. These kind of fixed stringent category, if you can do a 407 testing, then you can participate in our 407 study. Parth, anything else you want to add to it?
That we need to discuss with the center one-to-ones,
Yes.
and then we can take a call based on the finding or based on the feedback from the center. Like if you're fine with taking all ICU, GNB, CR isolate, or based on that, we can take a final call.
Yes. So what we encourage is we encourage all the centers who test for N407 to fill up the Google form, and then we'll individually discuss the criteria of testing 407 in your center.
13 centers have filled the
we'll
Google form
suggest
so
you
far.
some.
Okay, then only 13 centers have
So
filled the Google form for
yes.
407. For 407 study,
All those who do N407 testing enroll for it.
At present, I can see
anything. We'll discuss. Yes, 13.
So the center, we are encouraging the center, those who are testing with in 407 to participate in the study. Then we can discuss one-to-one and decide the rule, whichever is applicable.
So may I, sir? I have a very small basic question. We have been testing 407, and we have enrolled also for the subgroup study also. Only thing is, if the isolate is a sensitive isolate, it's not a CRO. So what our understanding was the fourth number point where you have written all carbapenem resistant isolates, CRE, CRAB or CRPA. That point is feasible. But if we select all blood isolates and then they are sensitive, what is the objective we are going to have for this particular study in that case? Like what exactly we want to address?
Yeah. So definitely your point is well taken. All blood isolate or all ICU isolate, if we take, then the sensitive isolates also we are doing 407, which is not required. So this is only a data collection which we are doing from the participating centers. After we obtain the information how they are using, no, we will form a common rule. And when the
Sir, if
common
you
rule is formed, more or less the common rule is going to be formed as CR isolate only.
Sir, if you allow 1 minute, what we have done is I have been using 407 since like I guess 1 year or more. So what I have found out is, and we have written this to the BioMero people also, that 407 card is actually not very good for the Indian setup anyway. We have only one drug there which is relevant, ceftazidime avibactam.
Yes.
Rest of the drugs are not relevant for our setup, except polymyxin. I agree if a clinician is interested. So we have in fact written a publication in process like that only, that 407 is actually not very useful for Indian setup anyway. And with
Yeah,
this solution coming up, ceftazidime avibactam can anyway be tested by Illusion now. So why to have ceftazidime avibactam alone in 407? That is, they can incorporate in 405 and 6 both like that for Pseudomonas if they want to, 6 may if they want that. But otherwise, it is just a wastage. So
But
I'm
by
anyway going
distribution,
to go for the study. But I think the objective has to be very clear if we are going for all isolates or all ICU. Otherwise, that fourth number point is relevant. That all CREs, CRAB and CRPA and everything we can assess. But otherwise, I don't think we will hit a dead end here.
I have the same feeling, Mirza. I also feel 407 is a redundant antibiotic card, because hardly anything which is useful now. CZA was the only useful drug, and now that we have to test separately, so hardly anything is there. Okay, I'll let everybody give their views, and then we will decide what to go about.
And one more thing, that by doing the distillation test, you won't be getting the MIC. So for that, you need to put up either by e-test, or you have to do this diffusion or use the
Yeah.
7
But that is what that first number study which we are going to do. But for the CZA alone, I don't know. I maybe you have some objective in mind. So then, and frankly, I have enrolled anyway. But I want to learn the objective, what exactly we are going to achieve with this particular.
Objective will discuss on that particular day when the individually the protocol will be discussed.
The other thing is like ampicillin sulbactam you are having in this 407, which is the drug of choice for CRAB.
Yeah, that is one extra thing, correct.
Yeah.
So Parth,
yes,
I request you to
pseudo.
call individual centers, okay? Not many have filled, only some 13 centers are there, and another
this
few days.
ceftolozane tazobactam again for CRPA. Yeah.
Yeah, so call individual centers and discuss the protocol part.
Okay.
Yeah.
So those are the extra things, information which you can get from all BLBLI combinations which are being there in the 407.
Yeah, so Ketan will go to the next study. So Dr. Sivanathan is going to be the regional coordinator for this study. So Siva, you are there?
Yes, sir, I am here, sir.
Yeah. So the first question what Sivanathan asked me, sir, if we are telling colistin susceptibility by CAT or CBD to all the centers to do, why to do a sub-study? What extra we are going to gain in the sub-study? So Siva, you are going to answer this question. After that, you discuss the protocol. Okay.
Sir, yes, sir. Sir, from this study, like what we are planning is like, it is more or less like what we are already doing, like Vitec we are doing, and as per the protocol from the VAMR phase 2, we are doing CBDE. So now what we are going to add one more step is colistin agar test. Though broth microdilution test is more preferable, but still we are trying to compare the colistin agar test with this other two methods. Only for the isolates, enterobacterials and Pseudomonas aeruginosa as per CLSI, where the breakpoints are available. And data entry will be obtained from separate Google spreadsheet. Like those who are going to enroll in the study, we will discuss a complete detailed protocol and the testing method. One thing I want to add in this protocol is, though colistin agar test looks difficult, but it is not that difficult, because we can perform at a time in single plate around 10 isolates we can test. So if a center is having a high load of isolates of enterobacterials and Pseudomonas aeruginosa, they can even daily test up to 10 isolate in single plate. Or even if a center is not getting frequent isolate also, they can at least perform once in a week, so that they can perform the colistin agar test like in single day in a week, or in once in a 10 days, if you are properly storing the isolate. So this is the only thing I can highlight, so that we can actually perform the test
so that we can get a data to compare all these three method, and we can get the results, so that we can able to see what basic difference is actually there. Why the micro broth dilution actually recommended from CLSI? Why the cholesterol agar test? Mainly the cholesterol agar test, like, not recommended for Acinetobacter. There are the major errors happened with the cholesterol agar test when they are doing Acinetobacter. So Acinetobacter we are not including in this study. All Enterobacterals and Pseudomonas we are including in the study. Sir, anything else to discuss, sir? I think basic thing itne hai. Like, those who are enrolling, then we can discuss a complete detailed protocol.
So, what we do in our routine VMR study is the participating center
CBD.
has to do a supplemental testing. Anyone
CBD
they
we are doing.
choose, they can do a broth macrodilution, they can do a CAT, they can do a C. Any doing one methodology to supplement the Vitek colistin results is acceptable for our routine VMR study. There are broth macrodilution. The best part is to compare broth macrodilution with your CAT to make sure that the CAT works. You compare your broth macrodilution with CBD to see if CBD has works or not, if he picks the colistin susceptibility properly. But what about CAT and CBD? How well they correlate among each other? Usually, most of the centers do either CAT or CBD based on their number of gram-negative isolate. If you have a high throughput lab, you get 5 or more isolates gram-negative. Easy to do a CAT, as sir very correctly suggested, because it's a spot inoculation. You can do multiple isolate testing on a single plate. If you are a low throughput system, 1 GNB, 2 GNB, 3 GNB in a day, then CBD may seem more appropriate for those centers. But what about CAT and CBD? How will they correlate among each other? What is the discrepancy between both are the recommended methods? CBD is also a recommended method. CAT also is a recommended method based on different principles. How well they correlate?
Okay, so which one will be taken as gold standard? Because when you compare CAT,
that's why in this study we are not taking gold standard. In this study we are simply comparing CAT results with CPD among each other. We are not comparing each of them individually to a. The best would be if we would have compared it with broth macrodilution, but that will put a very tremendous amount of infrastructural pressure to the participating centers. They have to do broth macrodilution and then do CAT and CBD. What we try to achieve in this study is just compare the result of CAT and CBD and see what is their discrepancy. How much the result interpretation of CAT and CBD varies among each other.
So, Ketan, but still we have to keep one thing as a standard. Like, which one you are going to keep a standard? That's the question. Like, when we designing the study also, that was my...
CAT and CBD, none of them can be taken as a gold standard.
Yes.
So BMD has to be the gold standard, Ketan. So if you want to compare CAT and CBD, which is better, so BMD has to be done. And BMD is, suppose out of 100 centers, let's say 5 centers are agreeing to do BMD. Then those 5 centers, we can compare CAT and CBD. And those 5 centers, we can do Acinetobacter, CAT and CBD also. And then we will see that why not CAT can be used for Acinetobacter. So that kind of analysis has to be done. So maybe you and Siva can discuss this once again, because without doing a gold standard CAT and CBD, how can be compared?
Yes, there are two ways to do it, sir. One objective is to find out which is more accurate, and another thing is what is the discrepancy. So these are two different objectives. Discrepancy can be found out without including a gold standard test, but accuracy can be included only with a gold standard test. So as you correctly told, that's why broth macrodilution will be keeping it as a plus minus. Those centers which will be including broth macrodilution also, or they may be doing it on a regular basis, for including those data only we can comment on the accuracy, which among CAT and CBD, which is more accurate. Discrepancy can be commented without a gold standard also. Like a CAT suggesting I and CBD suggesting R. How much is the discrepant results? In how many of the isolates we get a discrepant result between CAT and CBD?
So in this study, we will take centers, those who are ready to do CAT and CBD both. And also we will take those centers in addition to CAT and CBD. If they are doing BMD, we will take those centers also.
Yes,
right?
Yes.
So this is the plan, but it is little complicated. So the protocol has to be written in a flowchart method and should be shared. Then only the participating centers will understand the exact what we are going to do and they'll implement. Otherwise, many centers will wrongly feel that if you are doing CAT or CBD, anyone, we can enroll for this study. No. CAT or CBD, anyone is a part of our VMR study. You have to do either CAT or CBD. It is different that many centers have not started yet, but we are hoping that next 1 month, everybody will start doing either CAT or CBD. So Siva, you are going to take a session on CAT and CBD. When
Yes,
you will be taking that?
Sir, around 15th, sir, this month.
Yeah, so please fix a date. And
Yes, sir.
have you
Yes,
shared
sir.
the Colistin Agar the Sigma person's mobile number to all the participating centers?
Sir, not it, sir. Now I will share, sir. Like you have shared me, like so. But first we have to make a session, then we can share the number. I say that. So otherwise, I will share, sir, the number.
Yeah, so that they can discuss,
Yes, sir.
because we have
Yes, sir.
discussed with the vendor,
Yes, sir. Yes,
Sigma
sir.
company,
Yes, sir.
Bangalore company person we have discussed, and he has agreed to give 10% discount to
Yes,
all
sir.
the centers. So it will be around roughly 7,000 rupees plus GST.
Yes, sir.
Actual price is 8,200 plus GST. So somebody who wants to do CAT can easily procure from this company and start using, because in general the rule is, if your isolation load is very less, one isolate, one colistin testing per day, then CBD is appropriate. If your isolation, if your colistin testing is around 8 to 10 per day, then, or even more per day, 8 to 10 or even more per day, then colistin agar test is much convenient. CBD is going to be very active. So
Yes, sir.
both have their own advantage and disadvantage.
Disadvantage.
CBD for low volume center and colistin agar test for high volume center. So,
Sir, are you keeping BMD as a compulsory or optional?
If Ketan wants to see the discrepancy, so to see the discrepancy, the minimum compulsory thing is CAT plus CBD. Those centers who are doing CAT for CBD, both, they can enroll for the study. Among them, some of the centers who are willing to do BMD, we can do a another subgroup analysis where which is doing better. So it is little complicated. People may get confused. So you have to be very clear on the objective.
Hello, sir. I have one query. As a CAT, we are supposed to do on for all colistin testing. No? It's not about sensitive and resistant one. No?
No, yeah, yes. Enterobacterial and Pseudomonas, CAT or CBD, any one we have to do in addition to Vitek. Vitek result will be given, but we will do CAT or CBD, and then only the CAT and the CBD data only will be submitted to VMR. And
Sir,
yes.
And sir,
Yes.
for that Vibrio study, you will be considering cholera and then parahemolyticus, all the species of Vibrio, right?
Yes. Yeah, all species. Non-cholera Vibrio also.
Okay.
So that is why all specimen also. It's not that only stool, because non-cholera Vibrio can be isolated from blood or other specimens. So all specimens will
First
be isolated.
sample also.
Yeah, please ask.
Sir, please share me the CAT vendor, sir, so that we can purchase. We are from Andhra, sir.
Yeah, Siva, please post in the group.
Yes, sir. I will share the number. Yes, sir.
Yeah.
Yes, sir.
Yes, sir. Thank you, sir.
Sigma 2, two formulations are available. One comes as a 100 milligram box, and one comes as a 1 gram box. If you go with their what is
100
displayed as
milligram,
a
I have taken the code, Ketan.
yeah, but I want the individual centers to ask that representative about 1 gram also, because if you do CAT, then 1 gram would be the best to buy for you, because if you keep doing it on a regular basis, 100 milligram may give you a short amount. So if you're planning to start CAT as a routine, then and this will be cheaper. 1 gram is much cheaper as compared to 100 milligram. So
Okay, sir.
So you ask both the codes, and
No,
whichever
I
is
will,
cheaper in the
in
sense
that
like
case,
comparative relative rate is
in
cheaper.
that
It is
case,
not.
Ketan, I will ask the vendor the rate, because the individual centers, if they ask, he may tell the higher rate. I will ask the rate for 1 gram also. Okay.
Yes.
So I have posted in the VMR study phase 2 group the mobile number. His name is Mr. Vinith from Merck. The company name is Merck.
Okay, sir. Thank you, sir.
Yeah. So to all the centers, I would like to say that in study 6, whoever are willing to participate, BMD, if they want to do, it's not that BMD, in-house BMD you have to do. There are commercial BMD also which are available. Transisia commercial BMDs are available. So that is easily implementable. That is not very difficult. So
even high media is also having.
Yeah, high media also are now available. So whoever wants to do this study, please include BMD also in your protocol. So CAT, CBD, BMD. If you are doing all three together, then we can have a comparison nicely. let a very small number of centers participate, maybe 5 or 10. But if they're doing all these three, then we can include SNATO also, Ketan. When you include SNATO, because my objective is at the end of the study, I want to see SNATO can be rely on one of this test if you're not able to rely on Vitek.
Yes.
And one more objective is, like, we have asked all the centers to do CAT and CAT or CBD, anyone. So at the end of our study, also we will analyze all the centers are doing Vitek also. So compared to Vitek, CAT and CBD also they are doing. So how the Vitek is different than CAT and CBD, whether we can rely on Vitek data or not. So that also we will be able to find out at the end of our study. So these are the outcome which we are eagerly waiting for.
We'll make protocol accordingly, sir. We'll discuss with them and we'll make a protocol, sir. Yes, sir.
So first to collect the BMD interested center list,
Yes, sir.
CAT, BMD, CBD interested center list. Then you can discuss with them individually and then form the study protocol. Whether to do BMD optional or it is compulsory for all the subgroup. That you can plan.
Sure,
Okay.
sir. Sure, sir.
Okay, then any other questions are there before we close the meeting?
Sir, I have one.
Yeah, Ketan, you can remove the slide from the screen so that all the centers can on their videos. Whoever wants to speak can ask your queries, and all the participants are requested to on their videos if you are comfortable to on.
Yes, sir.
Yeah,
So my
please.
query is simple. So we have basically more or less 6 sub-studies now. So I am being the PI of VAMR study. And now we have different co-investigators for VMR phase 2 study anyway. For these sub-studies, because we are not going to have any MOU, can we have one particular specific coordinator for maybe one study each, or maybe 2 or 3 people who will look into along with me? Definitely, given those initial requirements were to told that the PI will be responsible and everything well taken. But for a helping hand, somebody along with me, can we have? So that because
That
that
is why
will
in the
be,
Google
otherwise it is
form
very
we have
hectic.
asked individual coordinator for every study, because we know that PI will be overburdened with so many small studies. So in the Google form we have asked individual coordinator for every study. So you can allot some of the faculty members for individual studies, and they will handle the studies, and you will be the overall responsibility you will take as a PI, but the individual studies can be handled by the individual coordinator. Similarly, for the project coordination sake, Ketan is going to handle all the 6 studies overall. So please do not ask any queries to me directly. You all are free to ask, you bombard your questions to Ketan. He is going to handle, because he has agreed that he will support me in this 6 sub studies. Because if individually I am going to address all these queries, even it is going to be very, very difficult for me. So let us empower everyone, because idea is to have the data available so that we can analyze, and we can say that we can proudly say that at the end of 2 years we will have a melioidosis antibiogram, Vibrio cholera antibiogram, hemophilus antibiogram. All these are dream come true studies for us. So to make it happen, we all have to join hand and empower each other. Any other queries?
So one more, Dr. Saraswati from SIC, sir. In the antibiogram list, can we add minocycline sulbactam for XDR isolates?
Minocycline sulbactam. That
Yes,
combination available?
Not available, sir, but our hospital treat with that MDR, XDR isolates, that the patients are getting cured. So this is...
Minocycline is...
maybe for crab they are trying that, I think.
Yeah, for crab they are trying.
Yes, sir. Not only for crab, for others also they are trying, and
So,
they are
madam, for one particular center, the study protocol changing is going to be very difficult. So
no, sir, just a suggestion. Are there any other center doing this, or anyone is interested? That's all.
Yeah, it will be difficult, because how minocycline sulbactam, there is no breakpoint available. So how you are going to how you are testing?
We are not testing, sir, but they are actually giving and treating with that, and they are getting good results.
That drug is available. That drug is available.
No,
Or
sir,
they buy minocycline
they buy
separately.
minocycline separately, and sulbactam separately, and they are giving to the patient.
But it is not available for testing, madam. I don't think sulbactam alone testing methodology or breakpoint is available. So once it is available only, we can do the analysis.
Okay.
Sir, I have one doubt. Ma'am, I have one doubt. What about you said in minocycline and sulbactam?
Yes, ma'am.
How they are
Yeah.
treating, ma'am? In
But
ICUs
they're
and all?
getting, yes, ICUs, they're getting separately, minocycline separately, sulbactam separately, and they are giving in
See,
combination.
sulbactam
They're
has
following.
anti-acinetobacter activity. So that is why they are combining minocycline and sulbactam. So for Acinetobacter XDR isolate, because nothing will be available. So this is one of the option what they're trying. It is good
Yeah, I
only,
think
but there is no testing
following
method.
ICMR guideline 2022. They have mentioned high dose sulbactam 9 gram along with other susceptible agent, maybe
No,
minocycline.
that for known Acinetobacter
For
infection.
known.
What about empirically using it? I don't think a valid reason of
Not
empirically.
empirically, they're not empirically using it, sir. Once it is like XDR, we don't have any other agent to test, then they are giving this.
Okay, okay.
Is it in
Only
high dose,
XDR
ma'am?
Acinetobacter.
Is it in high dose?
Yeah.
Ma'am, actually, I'm not aware of the dosing and all. The clinicians are doing, but they're getting good results. Clinical outcome is good.
Daily 3
So,
grams sulbactam is what they recommend. So I think they
3
will.
gram TDS, so it's 9 high dose.
Okay. Any other queries?
So one query we had.
Yeah. Hi, Dr. Tanya. Please ask your query.
So for the primary study, we have included sub-studies. We'll be able to include only the isolates from blood, no, sir?
Various substudies, various inclusion criteria. So, like for Vibrio, Haemophilus, Pseudomyeli, all these three, we just now we have discussed. We will include all specimens,
All specimens.
regardless whatever we have asked in the primary study. In this particular sub-study, we will include all specimens.
Okay, so would we require separate approval for that ethical clearance or something?
As far as study, as far as zipper is concerned, we do not request you for giving any separate approval. But if you are willing to do by yourself, then it is up to you to decide.
Okay, sir. Okay, thank you, sir.
Yeah.
Hello, sir.
Yeah.
Sir, we need any ethical approval for the sub-studies also? Ethical
No,
committee.
not required.
Okay, sir, another doubt, sir. We are entering into the IBAR data. So how we know that we are entering correct or not? Some drugs will be suppressed according to site infections. We are not suppressing right now. So as we are submitting whatever the VITEC was given, we are submitting the same thing into the IBAR. We should know whether we are submitting is right or wrong for the IBAR data, report data.
For antibigram, not all site-specific infections are to be suppressed. Some suppression is required that we'll be doing in the analysis part of antibigram. But the sample site-specific suppression is not recommended for antibigram preparation. So will not be needed for you.
Sir, actually, there is phosphomycin in pus sample and all, no, sir?
No, phosphomycin we are not taking, ma'am.
No, sir,
Phosphomycin
not
there is
for you. No,
Yeah.
sir, not for our study. We are asking like, is that we are going to report like we have to suppress and give the report?
Phosphomycin for the patient report you have to suppress because the VITEC is not the recommended method.
Yes, sir.
Therefore you have to suppress.
Sir,
And,
that's what we wanted to know. Like, is that we are submitting correctly or not to IBAR? So how can we know? Can you
can have
take
a
one
separate
question?
discussion with your regional coordinator. How you are submitting, you can show them so that they can guide you whether you are submitting correctly or not.
Okay, sir. Thank you.
Okay. Who is the regional coordinator, madam?
Mahalakshmi, sir.
Mahalakshmi, madam.
Yeah.
Okay, sir, thank
you. Yeah. Thank you.
So what is the ideal way to test phosphomycin then?
Phosphomycin.
Agar dilution.
Agar dilution.
Either you can do it screen agar, either you can do it. In general, for parenteral phosphomycin, it is agar dilution. Either you can do a screen agar testing, or you can do a cat-like limited MIC testing, or you can do a full-fledged agar dilution.
Ketan, think over obtaining a phosphomycin data because another sub-study you can plan. There are some participating centers who are doing phosphomycin. So, agar dilution
phosphomycin versus actual agar dilution of phosphomycin. This is a good comparative studies. We tell that it's not recommended because CLSI says that don't do it. What our data says, how much is the discrepancy is a very good study. Those centers which are willing to do agar dilution for phosphomycin can be a good study.
Dish diffusion not recommended, right?
Only urine isolates. This diffusion not for parenteral.
So that also can be included in the in the study objective. So a seventh subgroup study can be planned if you are interested.
We'll ask the centers with Google
Parth,
Form.
what you are
Let's see how
telling?
many
Is it what?
Yes.
Sir, there is one doubt, sir. Actually, for phosphomycin, hello, sir, am I audible?
Yes, madam.
Yeah,
Please tell.
sir, for urine isolates, we can do disc diffusion, no, sir?
Yeah, yeah.
Yes.
Okay. Yes, sir.
So Ketan, urine isolate diffusion and
E. coli
Vitek data
and
is
Enterococcus.
available for all isolate. Then only those centers who are ready to do colicin agar test, you can plan for a subgroup study.
Sir,
I
is
know
it that Vitek is giving pseudo, inflated positive sensitivity? Because we have a huge sensitive, say, enterobacteria, which are CRE also, but sensitive to phosphomycin.
What is your query, ma'am? Exactly, can you ask again?
Like Vitek is giving, huge sensitivity to phosphomycin. The CRE is all are also sensitive to phosphomycin. So,
that is
is
true.
it something wrong? Am I see that
That's what
Vitek
it
is
is not
giving?
reliable.
We're not reliable.
Because it is not reliable, we are not including in our
But in general, phosphomycin as a compound, it has a good susceptibility. But how much Vitek is reliable, we don't know. It's not that phosphomycin is a very useless drug. How it is going to be effective on CRA and XDRs? That's not the case. Phosphomycin, parental phosphomycin tested by broth microdilution has a good action on XDR, PDR isolates also. But Vitek reliability, we don't know until unless we look for it.
And even in terms of clinical benefit, if you think, then for a CRE or CRE isolate, phosphomycin, if it is also in vitro susceptible, it is not recommended because of presence of phosphate gene.
Yes.
So that is not recommended to use phosphomycin even for CR resistant, CR isolate. If you are getting susceptible result in Vitek, you are not supposed to use that, especially for Klebsiella and Pseudomonas isolates.
So do we have to suppress the result? It means?
Yes, madam. No, it will not come to IVAR at all because in our
No, sir, in general,
Yes.
In general, for the patient report, you should not report phosphomycin.
Thank you.
We have raised our concern to bioMerio, but bioMerio is not removing phosphomycin from the panel because ID physicians are using phosphomycin and they are pressurizing bioMerio to keep. They themselves don't have an answer how to rely the data. But yes, we can do the sub-study. How many of you often can agree to do a colicin agar test? If we have a little idea, at least some 5-10 centers are there, then we can really start doing the sub-studies. Any of you here are willing to do colicin, phosphomycin agar test? Again, for that you do buy phosphomycin powder. So maybe as a part of a PG dissertation or something can be planned. Suppose the 5-10 centers are ready to start, then we can discuss the protocol because we have a protocol in place. In JIPMER, the study has been done and we have a protocol in place.
So we can think on it. You can share the number like that sigma person, and we can compare the cost and can decide
Phosphomycin
on.
cost is how much, Ketan, when we had procured, you remember anything? Phosphomycin cost?
Don't remember, sir, but it was less monthly, I think.
2 years back, we have procured, I think. I will share the details. In the meanwhile, Ketan, you can discuss with your colleague in AIMS Patna, Dr. Gopi. When he was there in JIPMER, he has worked well on phosphomycin. So we can take his help also if required.
Yes.
Okay.
We'll create a Google form.
Sir, is it that phosphomycin should not be included in the antibiogram? A hospital antibiogram?
Yes, madam.
On the basis of Vitek
On the
MIC?
basis of Vitek, you should not include.
So what about urinary isolates?
Urinary isolate, if you are doing a disc diffusion separately, then you can analyze. But on the basis of Vitek, even on urinary isolate, you should not declare the antibiogram.
Thank you, sir.
So looks like many centers are interested for phosphomycin, but only thing is they don't know whether phosphomycin agar test they can be able to implement or not. So if they are ready for it, then we can go for another substudy.
No, sir, but every time Vitek result, we are giving, it's coming as sensitive and we are releasing the report.
Yes, ma'am. If we do this agar test comparison with Vitek, no, we will do only for 3 months. 3 months ka data analysis. Once we will implement this, we will see that if the Vitek data is reliable, then we have a proof, and based upon that proof, we can continue to use Vitek.
Yes, sir.
So it is definitely very useful sub-study. Or if the result of our study will say that the Vitek data is not reliable, then we can loudly tell to the audience that we should not use fosfomycin.
Sir, what about the cost and vendor, sir, for fosfomycin?
I will find out, ma'am, and then I will post in the group. Anyway, we have 10,000-15,000 odd we have spent, I think, Ketan. Exactly, I
Yes,
don't
yeah, somewhere around less than 15,000. Fosfomycin is cheaper than colistin as a drug powder. Much cheaper. We'll share the details, ma'am.
Okay.
Yeah.
Thank you, sir.
Thank you so much. I mean, with this discussion, we have brought another study in our mind. Okay, then, if no other queries, I request all of you to fill the Google form. If you would have already filled the Google form and you want to enroll for some other studies, then you can refill the Google form. Okay. And as we have discussed now, Haemophilus, Pseudomyeli, all that, Vibrio, all that, all of you can enroll because there is there's no restriction for that. You're not doing anything extra for that. So please wholeheartedly participate in those subgroup studies. The maximum response we got for ceftazidime avibactam aztreonam study. So Parth, congratulations for you. So you are going to monitor the study and please do nicely. Okay. Make sure that everybody is adhering to the protocol.